My topics I have to cover are resistance, but also kinase inhibitors for rare mutations, and I think one of the most interesting topics is also on patients who got combination checkpoint blockade and patients who discontinued from treatment. How do they do in terms of safety and efficacy? Understanding resistance obviously will open some ways to get around it, possibly prevent it. On the other hand, if there is a molecular basis for resistance and there are certain indications, for example, the checkpoint blockade might be dependent on the interferon signaling pathway, and if there are mutations or epigenetic modifications, it might be possible that it's not so easy to overcome or to prevent. The same with the targeted therapies. We know that certain amplification and mutations of certain key signaling molecules do occur, and I think the only way to prevent that is to hit hard and early in order to overcome and possibly delay resistance. There are multiple ways to overcome resistance. I think key is to understand resistance. We know, for example, for checkpoint blockade that obviously the interferon signaling pathway is of critical importance, and mutations or epigenetic modifications cannot easily prevent it. The same if we analyze the MAP kinase pathway in the context of BRAF/MEK inhibition. Also here we see mutations, amplifications, which can be in part prevented by combined approaches. I think in summary is that we need to combine approaches, hit hard and early, and possibly also to combine targeted and immuno-oncology approaches. Rare mutation subgroups is obviously more challenging. I think we have learned by the sequencing approaches that there are rare mutations. The problem is that there will never be firm clinical evidence how to treat these rare mutations, because we will have, on one hand, preclinical science supporting that there is resistance or sensitivity to certain drugs, but and there will be possibly also some patients' case reports or small series of patients treated with certain drugs, but there will be never a clinical trial supporting really clearly the clinical evidence and the magnitude of the evidence. So that's challenging. It's more based on preclinical data, drugs which are available, testing in the lab, and then testing efficacy in the clinical setting. I think one very nice example is, for example, the experience we have with c-KIT. This is one of these rare mutations which we know nowadays occurs in acral melanoma, in mucosal melanoma, and in chronically sun-damaged melanomas. Roughly 10% of these already rare subgroups, and this is not one mutation like the BRAF mutation. We know that c-KIT mutations occur in various forms, and also amplification of c-KIT has been seen, and all the trials which have been done with c-KIT have included most of the mutations or also amplification. And there is some early indication now that amplification obviously as a sum do not respond to inhibitors like imatinib or nilotinib. On the other hand, if you analyze the different mutations, which are there, and their sensitivity to drug exposure, there is a clear tendency that only 2 or 3 of these mutations really seem to be sensitive in a range between 20 to 50%. So that's already an indication, but it's a solution only for a very small fraction of our patients. One very interesting piece which was presented here is the experience of patients who have been treated with nivolumab and ipilimumab, the PD-1 antibody in combination with the CTLA-4 antibody. This regimen is already approved in Europe and the US and has been tested in 2 larger trials, the Checkmate 69 and the Checkmate 67 study. One of the major challenges of this treatment approach combining checkpoint inhibitors is toxicity. More than 50% of these patients will experience grade 3 or 4 toxicity, and almost 40% of the patients need to discontinue treatment early. So the question which was asked is, how is the efficacy of this combination in the patient group who has discontinued, and how does that compare to the patient group who is continuing and staying on treatment? And if one looks on this meta-analysis now, which was presented, more than 400 patients were included, is that the group who has discontinued, a group of more than 170 patients, stopped treatment quite early in the range of 1.5 months. And the group who stayed on treatment had more than 9 months of treatment. So that's already a substantial difference. So if we look now on the efficacy, then the surprise is actually quite large, because the efficacy in terms of tumor control measured by progression-free survival time is better in the group who has discontinued early. It's more, in median, 10 months for the patient group who continues versus more than 16 months in the patient group who has discontinued early. This is statistically highly significant. And also, if we look at response rate, the response rate is respectable 50% for the patient group who has discontinued, but is 68% for the patient group who has discontinued early. So there is clear efficacy. So if we now look, what is also of great importance, durability of response. The durability of response is not reached. The follow-up time is more than 20 months. And so usually the response, the clinical response is achieved within 8 weeks. So for the first staging, you see that the patient is benefiting. But we know after 1 year, if we look at the patient groups, 90% of the patients who had discontinued early still do not need any treatment. And this is in comparison to patients who have continued treatment, close to 80%. So the only difference in favor of the group who has continued treatment is the duration of response, ongoing responses. So possibly for longer duration of response, you need some more clinical treatment. So it's currently open what to do with these results. I think the caveat is it's a retrospective analysis of a large group of patients. There is clear evidence that the patients who discontinue seem to benefit. Toxicity in these patients is much, much higher. The burden of toxicity, almost 50% of the patient who had to discontinue had 2 or more organ sites involved by the autoimmunity, in contrast to 80% of the patients who continued treatment had either no toxicity or only one organ system involved. And this is also visualized by the fact that the patients who discontinued needed much more immunosuppressive agents like infliximab, 10% in that group, 1% in the other group. So this is quite interesting result, but poses obviously a lot of questions now what to do. Should we treat patients with a combination only for 8 to 12 weeks? Or is this toxicity, this severe toxicity and the good clinical response just an indicator, some sort of a biomarker for a particularly sensitive patient group? So I think that needs to be addressed in a prospective study.